Okay. Here we go. Well, we could get started and then more people are going to
Yeah.
hop on here. Well, good morning, everybody, for the last Myeloid Network seminar of the year. I'm really pleased to introduce our speaker today, who is a talented associate professor at Harvard University. He's Dr. Andrew Lane, is uh, the physician in the adult leukemia program and investigator at the Department of Molecular Oncology at the Dana-Farber Cancer Institute. He's the associate chief in the division of hematologic neoplasia and the director of the Blastic Plasmacytoid Dendritic Cell Neoplasm Center and co-leader of the Dana-Farber Harvard Cancer Center Leukemia Program. He focuses on new therapeutics for high-risk leukemia and lymphomas and has developed a number of therapeutics for AML, MDS, and BPDCN. So he's going to tell us today about his work on a new therapy for AML, uh, which is the PI3 kinase gamma inhibitor, eganelisib. So Annie, thank you so much for joining us, and we look forward to hearing about your work.
Thank you very much, Judy, Jen, Jelani, everyone, for the invitation. I'm going to share my screen, which we tested a moment ago, so we know it's going to work. Okay, hopefully that looks good. If anybody wants to jump in at any time, please do so. But I definitely will have some time at the end for questions as well. And I'm going to tell you about this story that Judy knows very well. One, because she's the OG expert on this pathway that we were inspired by many of her papers when we were working on this project. So I'm going to take you through a little bit of what we recently published and then also some new data about new directions of how we're taking this to the clinic and what we're still working on in the lab. Now, I know that most of you think about myeloid cells in the tumor microenvironment, I believe, and in other contexts, but I actually think about treating myeloid diseases. And so this is a story about AML and related myeloid diseases where we came upon PI3 kinase gamma as an interesting target. The only relevant disclosure is that I'm the co-founder of a company now that's trying to take this therapy into the clinic. And so you may discount everything that I say as potentially biased. Okay, so for those of you who don't think about AML, I'm going to give you the 2-slide primer on how we think about this disease. So back in the 1970s, the FAB, the French American British classification of AML, came up with this M0 through M7 classification, where basically we classified AML by how it looked under the microscope. And that was really all we needed to do because we didn't treat any of these leukemias any differently. They got high-dose chemotherapy in a younger person who could tolerate it, and we tried to get patients to a stem cell transplant. And it didn't really matter that there were these different types of leukemia, although they did seem to have different clinical characteristics based on the amount and the type of differentiation of the leukemia cells. So for example, leukemia without much differentiation or leukemia with myelomonocytic or monocytic differentiation or erythroid differentiation. This has kind of gone out the window now because we have genomics, and AML really was the poster cancer for understanding cancer genetics. And it's because it's relatively easy to get samples of the tumor, and we can sample it longitudinally through therapy. And so this is the world of AML genomics, actually from a few years ago now. It hasn't changed too much, but it's really complex. And AML, if you look at this, is probably 20 or 50 or 100 different subtypes where there's both chromosomal rearrangements and point mutations. And that is good in understanding the disease, but it's actually becoming challenging for drug development because, if you have a drug that targets one of these point mutations, it may exist in the context of many other point mutations. It may exist in the context of different chromosomal backgrounds. And it becomes harder and harder to sort of develop a drug in a very small subset of patients. But that has happened. Some of these mutations with the stars on them here in 2025 actually now have approved drugs, but it's still the minority of AML patients. And so we were thinking about AML from back in the perspective of the prior slide of sort of the differentiation state of leukemias, and could that tell us anything about the disease? And here's sort of a simplified schematic of hematopoiesis, including, primarily myeloid development here to mature cells like dendritic cells, monocytes, granulocytes, and here's lymphoid progenitors over here coming in. And we really were interested in this because, as Judy mentioned, in my lab, we're interested in this rare, very rare subtype of leukemia that's thought to be most closely related to AML of dendritic cells, essentially a leukemia of these plasmacytoid dendritic cells. Although there's a lot of functional and clinical and genetic overlap between that leukemia and AML and even a little bit ALL. So we said, let's take a step back and maybe we could study these dendritic cell leukemias, find some dependencies that are shared with more common leukemias like AML and ALL, and that might lead us to something that would kind of take us out of the box of having to go down to these tiny slices of genetic background. So that was how this started. And Qingyu Lo, who's a super talented now instructor in my lab, took this on with the hypothesis that he could find a shared subset of leukemias that have common dependencies. And particularly interesting on the AML side is that BPDCN has the most overlap sort of clinically, genetically with monocytic AML. And that's a clinically important patient population because venetoclax, which is a BCL2 inhibitor, has really kind of revolutionized AML care. It allows us to give effective therapies to older patients who can't tolerate intensive chemotherapy, for example. But the monocytic AMLs remain a problem because they are the least sensitive to venetoclax up front. And when AMLs relapse, they often grow out on venetoclax with monocytic clones. So this was kind of the idea. Can we look at these cells and figure out some commonalities? But we'll come back to what I found, but I like at this point to introduce PI3 kinase. I guarantee that many of you in this audience are much more expert at PI3 kinase and signaling pathways than I am. Certainly Judy is, and many others. But what is important to think about, and I think in the world of heme malignancies, when you say PI3 kinase inhibitors, most people think about the problems we've had developing them in CLL. So it turns out that there are class 1A and class 1B PI3 kinases. I think most of us think about, when they think about PI3 kinase, we're thinking about the class 1A. That's the alpha, beta, delta. That's the kinase subunits, P110, alpha, beta, and delta. And in the cancer world, this is very well studied. There are multiple drugs that have been developed, including approved drugs. For example, PI3 kinase alpha inhibitor alpelisib, approved for PI3 kinase PIK3CA mutated breast cancer. In CLL, the target of interest is delta because that's a dependency in those cells. And there have been multiple drugs developed. However, they've sort of fallen out of favor because of autoimmune toxicities that come from targeting delta in T cells. PI3 kinase gamma is definitely much less studied. It's really exclusively expressed in hematopoietic cells. And it actually has somewhat of a different signaling pathway. We'll talk about a little bit about what we found, but definitely these G protein coupled receptors are sort of the primary upstream stimulus to PI3 kinase gamma, although other more traditional RTKs, RAS, also interact with the gamma pathway. And it really wasn't previously known as an intrinsic cancer dependency. What it was known as, based on Judy's work, is that it is important in tumor-associated macrophage polarization, and that by inhibiting PI3 kinase gamma, you can repolarize tumor-associated macrophages to make them more anti-tumor and synergize with PD-1 blockade. And because of that, gamma inhibitors were in development in solid tumors, including one that we'll talk about more that made it to phase 2 trials in combination with PD-1 across multiple solid tumors. Okay, so Qingyu did a very straightforward CRISPR inhibitor screen in collaboration with the Broad Institute, first in BPDCN cells, just looking for dependencies. This disease was not included in the DepMap cancer cell encyclopedia, so we thought it was low-hanging fruit to just figure out what are the dependencies of BPDCN leukemia cells. And the screen worked very well. We could block out different groups. We got rid of the known essential genes and found these core dependencies of BPDCN cells. We started with one cell line, looked across then multiple. The core dependencies overlap with gene sets that have been implicated in AML. That works well because that tells us what we knew, that this disease is probably most closely related to AML. And then the essential genes hit essential pathways as expected. One of our top hits was a gene called PIK3R5. PIK3R5 encodes P101, which is an obligate heterodimer complex with the kinase subunit P110 gamma. And here's another cartoon showing you how PI3 kinase gamma is slightly different than the class 1A PI3 kinases. And here in the middle are data from our screen showing that really none of the either regulatory or catalytic subunits of the class 1A PI3 kinases were dependencies in our cells, but both PIK3R5 and PIK3CG, the kinase module, were important. On the right is an analysis from another paper that came out a few months after ours, which we'll talk about again later, which also kind of struck us as why this might be interesting. So this is an analysis of the Cancer Genome Atlas. These acronyms here are different cancer types. And what you can see is that in AML, there is expression of PIK3CG, the kinase, and it's really not in too many other cancer types, maybe in lymphoma. And that the PIK3R5 is also expressed. And importantly, there's a differential expression between the normal tissue and the tumor tissue in the AML analysis here of that PIK3R5, which was our top hit. So we said, okay, this looks kind of interesting. Then we went to DepMap. We do lots of CRISPR screens, so we always go look at DepMap to see where things fall out across cancer cell lines. And this kind of solidified that we were onto something that might be interesting. So these are DepMap data for PIK3R5 and PIK3CG. Each dot is a different cancer cell line, organized here by sort of tissue of origin. The further to the left on these graphs are cells that are dependent on that gene. And you can see that both PIK3R5 and PIK3CG have just a minority of cell lines that look like they have dependency, but they're in blood cancer cells, both lymphoid and myeloid cell lines. And then interestingly, if you look on individual cells, it's the same cells that are dependent on each of these two proteins that bind to each other in the signaling complex. So this is not some kind of artifact. There's something really going on here, that there's a selective dependency in blood cells. The other reason we decided to go forward with this was that there was a drug that we could use in the lab and in animal models and maybe even in patients. So here's the summary of a ton of work. Here's from Judy's lab, 2 high-profile papers 10 years ago showing what I mentioned earlier, that PI3 kinase gamma is important in tumor-infiltrating myeloid cells. You all in the myeloid network probably know this already. And there's a drug called eganelisib, which was originally being developed by Infinity Pharmaceuticals, which is about 100-fold selective for gamma over the class 1A PI3 kinases. And that is important because it avoids the toxicity, that autoimmune toxicity of delta inhibition. And it's an orally bioavailable, 1 pill a day, continuous daily dosing, again, well tolerated in phase 1 studies. And it had advanced as far as this MARIO-1 trial, and in fact, actually some other trials that were not yet published, not sure if they will be, in solid tumor patients where eganelisib was combined with either PD-1 blockade or chemotherapy or both in solid tumor patients. And again, it was tolerated, but there wasn't overwhelming evidence that there was a sort of synergy by the combination with PD-1, although the company was never able to get to a phase 3 trial where they could really ask that question. So we'll come back to the drug. So in the lab, we did a number of validation studies. I'll just summarize them here for you. We moved from CRISPR to SH. We did CRISPR knockout in addition to CRISPR-I, and we validated the findings of the study. We also moved to a larger panel of cell lines across AML, actually, and ALL. And essentially what we found is that the higher level of PIK3R5, there was more PI3 kinase gamma dependency. And we could test that with genetic loss, and we could test that with drugs. So here's eganelisib in orange, and we can see that the cells with sort of the highest dependency on the gene both the regulatory subunit and the kinase are most dependent on the gamma selective inhibitor. They're also dependent, they're sensitive to this duvelisib, which is a delta gamma inhibitor, though this is about 10-fold more delta than gamma. And some AML cell lines are sensitive to this pan class 1A PI3 kinase inhibitor, but it's not concordant at all with the PI3 kinase gamma. So this really, we think, PIK3R5 and PIK3CG are markers of PI3 kinase gamma dependency. We went to primary cells using a patient-derived xenograft bank that we built a decade ago here at Dana-Farber, and luckily I kind of house in my lab. And what we have are PDXs that grow in immunodeficient mice across multiple heme malignancy subtypes. And we also have characterization of those PDXs by RNA-seq, DNA sequencing, et cetera. So we could go and pick out PDXs across multiple types of leukemia and stratify them into PIK3R5 high and low. And when treating those leukemias in vitro with eganelisib, we saw that the higher PIK3R5 PDXs were more sensitive to the drug. In vitro treatment or ex vivo treatment of AML with drugs in the lab is very difficult. Basically, all the cells are dying. They don't grow. They don't proliferate. They don't live for more than 3 to 5 days. But you can see a difference, at least in viability, in drug sensitivity. So this was all encouraging. We did a little bit of work to try to understand more about what are these leukemias that are sensitive to PI3 kinase gamma inhibition. And so if we did gene set enrichment analysis on across histologies, actually, PIK3R5 high versus low, we got all of these signaling sort of gene signatures for cytokine signaling, inflammation. And this was also interesting to us because we know that AML kind of lives in an inflammatory microenvironment and even thrives in an inflammatory microenvironment. And particularly these monocytic type AMLs are associated with that kind of microenvironment. We'll come back to that a little bit later. And then we were able to make what we called an IIRS, innate immune response signature. This is sort of a 30 gene signature that includes PIK3R5 and then a number of other of these sort of cytokine signaling, toll-like receptor signaling pathway genes that was a little bit better actually at distinguishing the likely sensitive leukemias from not. So now we had a signature that we could go after. We've done a little bit of work on this sort of upstream signaling, and it turns out that you can actually increase the level of PIK3R5, not so much PIK3CG, by exogenous inflammatory stimulation. So resiquimod is a toll-like receptor agonist. We saw similar phenotypes with interferon, and that fit with those gene expression signatures that we saw. It could actually also sensitize sort of moderately sensitive PDXs to become more sensitive to eganelisib by treatment with these inflammatory agents. And so resiquimod here increases PIK3R5 and increases the sensitivity of those leukemias to eganelisib. I'm not going to show it here. Chingy worked out the biochemistry of this, and actually this is something that's already known in the class 1A PI3 kinases for sure, that the level of these regulatory subunits, the non-kinase subunit, like the PIK3R5 here, the higher the level, the more activity of the pathway. It sort of drives activity of the pathway and vice versa. So it made sense to us that if we have stimulation that can increase this regulatory subunit, it might increase activity of the pathway and possibly then increase the dependency on eganelisib. So in the leukemia world, based on that pie chart I showed you earlier, everyone will now ask at this point in the presentation, which leukemia patients are these that are going to be most sensitive to inhibition of this pathway? Because we do DNA sequencing on all patients with AML immediately at the time of diagnosis. We do it with rapid panels to get the data back within a few days. And it would be amazing if you could just select a genetic subgroup for treatment with your new drug. It turns out that's not so easy with this pathway. So it is true that the patients with higher levels of PIK3R5 or higher levels of this immune response signature actually have poorer prognosis overall. We even did multivariable analysis to show that this is indeed independently associated with overall survival. That raises, more interest. However, when we look at sort of the most commonly mutated genes in AML, there's really not an obvious segregation of certain genetics here. Now, in larger and more recent data sets, we can talk about this later, it does look like there's a trend toward an association with RAS pathway mutations. NRAS, KRAS are what are common in AML. And that is interesting because of the monocytic dendritic phenotype is also associated with NRAS, KRAS. So there may be a way to enrich by genetics, but it's not, definitive. But here we do have that FAB subtyping for these patients. And again, not absolute, but the patients with M4 and M5 subtypes, those are the ones with monocytic differentiation, clearly are enriched for the high signature. And so that at least gives us something that we can kind of go after. And again, is interesting to the leukemia clinicians because that's the subset that is not benefiting very much from venetoclax. This is kind of a cool experiment that Qingyu came up with. I think this is kind of brilliant. We wanted to figure out what's upstream regulating PIK3R5, after the inflammatory stimulation. And he did ATAC-seq in primary leukemias that were either high or low for PIK3R5. So red are high, blue are low across different subtypes, BPDCN, AML, even B-cell ALL. And as you'd expect, the leukemias with higher expression have more accessible chromatin at their transcription start site. What's cool is that he then overlaid data for transcription factor binding sites within that ATAC-seq peak with his CRISPR screen and saw that there were just two transcription factors that bind in that peak that were also dependencies in the same cells that were dependent on PI3 kinase gamma, this ASH2L and SPI1 or PU.1. And so then he tested those two individually in the same cells and showed that SPI1, but not ASH2L knockdown, decreases PIK3R5 expression and blocks the ability of those cells to respond to the inflammatory stimulation upstream. So we think that this goes sort of inflammatory stimulation through PU.1 transcription factor to drive PIK3R5 expression. And that's, maybe if you're a myeloid person, you say, well, that's actually pretty obvious. PU.1 is just a well-known transcription factor that drives these monocytic differentiation, particularly in AML and in normal biology. So that it kind of makes sense. So what's downstream? And this is where there's been quite a lot of discussion. I wouldn't say the word controversy, but I will walk you through why this was sort of unexpected, what we found might be signaling downstream. So in the most sensitive, most PI3 kinase gamma dependent cell lines, when we did RNA-seq, either after drug treatment or CRISPR-I knockdown of PIK3R5, we didn't see a strong signal for downregulation of PI3 kinase AKT-mTOR. But this is with genes that are in common between these two things. So genes that are commonly changed with eganelisib and PIK3R5 knockdown. We'll see why that's important on the next slide. But so we were like, well, maybe there's something else going on. So we did proteomics and phosphoproteomics. So proteomics showed almost nothing significant at short time points after either drug treatment or genetic knockdown, except for PIK3R5 and PIK3CG themselves. But at the phosphoproteome, of course, there were a number of changes, and we were particularly struck by this PAC1 phosphorylation change. These labeled on these two graphs are the phosphosites that are in common between both drug treatment and genetic knockdown. What you do not see here is AKT phosphorylation. It is here in the drug treatment, but it is not seen in the genetic knockdown. I'll show you the Western blots on the next slide. But why is PAC1 of interest? So PAC1 is this P21 associated kinase. It's downstream of a lot of oncogenic signals, been associated with like cytoskeletal rearrangement, but also RAS signaling and others. And it's actually been implicated in AML. Uli Steidl 10 years ago had a series of papers about PAC1 dependency in AML. And companies actually tried to make PAC1 inhibitors, but they're super toxic because it inhibits lots of essential pathways. But this was it piqued our interest. Okay, there's a ton of data here, but I'm going to walk you through what I think is interesting about this and interesting for those of you who are doing both genetic manipulation and chemical manipulation, you don't always get the same results. And I think it's important to look at both. Okay, so on the left are cells that are very dependent on PI3 kinase gamma with non-targeting control guides, PIK3R5 CRISPR-I or PIK3CG CRISPR-I. Okay, so the CRISPR-I works and you get loss of phospho-PAC1 specifically at this S144 site, just like we saw in the phosphoproteomics. AKT at this S473 site, which is important downstream of PI3 kinase signaling, not only does not go down with knockdown, it actually goes up, which we think is from some compensatory feedback pathway, whether it's from PI3 kinase delta or another PI3 kinase. I don't know that we know, but it actually goes up. In contrast, on here in the middle is drug treatment. So this is DMSO, eganelisib, or LY29, which is the pan PI3 kinase alpha inhibitor. So with eganelisib, phospho-PAC1 goes down in 2 different sensitive cell lines. And LY29 does not affect phospho-PAC1. But if you look down here at AKT, phospho-AKT is suppressed by eganelisib in the sensitive cell lines, as well as by the class 1A PI3 kinase. So I think multiple PI3 kinases may be converging on these substrates, particularly phospho-AKT, but PAC1 might be uniquely downstream of PI3 kinase gamma here. And that fits with effect on cell growth. So the pan PI3 kinase class 1A inhibitor inhibits phospho-AKT, but does not affect growth of these cells, whereas the gamma does. And so we took that one step further over here with these FRAX drugs are PAC1 inhibitors, and the MK is an AKT inhibitor. And the PAC1 inhibitors inhibit PAC1 phosphorylation, but not AKT phosphorylation. They affect growth of the cells. The AKT inhibitor does not affect PAC1, does affect phospho-AKT, does not affect growth of the cells. So I think this is not totally settled because, what we're going to give to patients is a drug, an inhibitor, not a genetic knockdown. But I think it just shows you that there can be differences between, clean loss of one kinase subunit and a drug that probably is hitting gamma, delta, and, maybe other PI3 kinases. Okay, what else is downstream? In the leukemia world, mitochondrial metabolism is important, particularly, for 2 reasons. One, unlike other cancers, the Warburg effect, et cetera, where cancers become more reliant on glycolysis, in leukemia, actually, AML cells are very dependent on OXPHOS and not glycolysis. And that's also important because venetoclax, this drug that now just about every patient with AML gets treated with, is particularly active in cells that have a dependency on mitochondrial oxidative phosphorylation. And so it was quite interesting for us to see that PI3 kinase gamma knockdown or inhibitor treatment inhibits OXPHOS, but not glycolysis in these AML cells. Again, supporting why it might be particularly effective in AML. And so before we went into animal experiments, we wanted to test both single agent and combinations. And we tested a number of drugs that are active in AML. We kind of threw things at the wall and saw what was most synergistic. And the most synergistic 2 drugs were cytarabine, Ara-C, which is the most frequently given standard chemotherapy in AML, and venetoclax. And, at this point, when we were working on our project in somewhat isolation, not knowing that other people were working on this pathway in AML as well, we said, this might be the case? Well, it's known that in post-chemotherapy persistent AML cells after Ara-C treatment, there's a known activation of GPCR survival signaling. And GPCR survival signaling, as I said, converges on PI3 kinase gamma. So maybe that's why there could be synergy. And with venetoclax, I just mentioned the OXPHOS, and that's known that sort of things that inhibit OXPHOS are known to synergize with venetoclax and AML cells. So that also made a lot of sense. So we decided to go forward with cytarabine because we thought it was actually more synergistic, and it was kind of an interesting combination that not a lot of people were using cytarabine combinations in AML. Every paper is using some kind of venetoclax combination. So we went first into a subcutaneous model where we could harvest the tumor cells and do additional PD work. And so this is a subQ model with these BPDCN cells that we made from a patient, a PDX. treated with either eganelisib alone, cytarabine alone, or both. You can clearly see that there's at least an additive effect of the combination of both, and both single agents have effect. We also saw in vivo inhibition of phospho-PAC1 at this S144 site with eganelisib and with the combination. And then, kind of really to take this home, we have this PDX collection that grows as intravenously injected AML or ALL or BPDCN in NSG mice. And Qingyu selected 3 leukemias that had very high PIK3R5. I think these are in the top 10% across our cohort. And so you predict that they have single agent sensitivity to eganelisib, and they do. That's the black versus the orange line in all cases. Cytarabine has some single agent effect in these leukemias, and the combination was at least additive, if not synergistic. So that was great. I think we could have just stopped right there, but I said, it would be really cool, Qingyu, if you did the negative control as well to prove that your biomarker might be useful. And so go find some, same histology, but leukemias that have very low PIK3R5. And so when he did that in some on that lowest 10% PIK3R5 expression, the black versus the orange, no effect, essentially, of the eganelisib single agent as predicted. But we were shocked by this really sort of incredible synergy with cytarabine that you saw in these low leukemias. And I'm not sure we completely know why this is the case yet, but we followed up on this prior publication that I just mentioned about persistent cells after cytarabine activating GPCR signaling. And so Qingyu took these PDXs and treated them with Ara-C alone and collected the MRD cells, if you will, the residual disease cells after cytarabine, and did RNA-seq and some Westerns. And what you can see is that, as had been reported previously, the persistent cells have activation of pathways of GPCR signaling, particularly via these purinergic receptors. And phospho-PAC1, again, a marker that's been associated in our hands with sensitivity to PI3 kinase gamma, is actually elevated in the persistent cells compared to the untreated. So this gave us a possible reason why there might be this particular synergy with cytarabine. Okay, so we were pretty excited about this, trying to think about how we're going to move this forward. And then to our surprise, 2 other papers came out a couple months after our paper. And in fact, during the sort of process of our paper being reviewed, I heard about both of these other papers. And what was very cool is that they all converged on the same conclusion that AML has a particular dependency on PI3 kinase gamma, but the other 2 groups did it by completely different approaches. So ours on the top, this is from a review. I like this picture because Qingyu and Amanda Fortune in my lab are much better at making graphics than I am. And I think this is very pretty. So this was our story on the top. We used CRISPR screening to look for dependencies in AML. A group actually here at Dana-Farber, believe it or not, that I had no idea was working on this project in collaboration with Judy, was looking at a mouse model of AML and had compared AML stem cells versus normal stem cells and looking for differential dependencies, landed on PI3 kinase gamma. And then the third group, which was from Duke and University of Paris, I showed one of their figures earlier. They were looking at TCGA data for pathways that were both leukemia specific and that seemed to be more differentially dependent in tumor cells versus normal cells. They also landed on PI3 kinase gamma. Here are the 2 other papers and a couple of interesting things about these 2 papers. So this paper in Blood actually gives us, I think, a better explanation for why there might be particular synergy with cytarabine. So in this paper, they did a lot of metabolic profiling of the AML stem cells versus normal stem cells. And they showed that inhibition of PI3 kinase gamma in the AML stem cells leads to inhibition of this pentose phosphate pathway. And the consequence of that is that it depletes the nucleotide pool from those cells. And it has been known for decades that the level of DCTP in a cell determines the sensitivity of that AML cell to cytarabine, that Ara-C. That's the C in Ara-C, is CTP. And so that makes a lot of sense as to why we're seeing this remarkable synergy with cytarabine. They also used eganelisib, and it worked in their mouse model. Second group as I mentioned, used the TCGA. They also used eganelisib and showed strong synergy with venetoclax, including in vivo, which helps us as we move for clinical development because we didn't pursue the venetoclax, but we now have other in vivo data. Now they said that the eganelisib wasn't as good as they would like. I agree with them. I think there are some drug-like properties that could be better. So they actually developed a PI3 kinase gamma degrader and tested that in these models and showed that it was as good, if not better, than eganelisib in some model systems. We've since been testing that, and in our hands, it's actually kind of similar to eganelisib. Okay, so what have we been doing since then? So we want to try to figure out how to get this idea to the clinic. We want to understand the mechanism even better. And to get to the clinic, it would be helpful if we could select leukemia patients that would be more likely to respond to the drug. So we've started with what we know, and these are 23 AML primary samples just randomly chosen out of our tissue bank. These were cryopreserved bone marrow specimens, and in no particular order or categorization, we made Western blots and we tested treatment with eganelisib in vitro. And these color scheme and categorization is somewhat arbitrary, but we tried to divide them into three groups. The ones that were PIK3R5 low or high, and then amongst those that were PIK3R5 high, whether or not we could detect phospho-PAK1. Again, this is sort of a crude method, but it gave us some separation that I think is interesting. And we compared this to normal, healthy donor human CD34 positive cells as a control. And I think of this as kind of a third, a third, a third, that across AMLs, as far as I can tell so far, there's a third of patients that seem to have low PIK3R5. They don't really respond to eganelisib as a single agent. And then amongst the high PIK3R5, they seem to respond better. And you can even subdivide those possibly by the ones that have phospho-PAK1 as being even more sensitive. So this helps us as we're starting to kind of frame around what we're going to do clinically. So as I mentioned, this company Infinity had been developing this drug, and I was talking to people at Infinity. And then to my chagrin, I learned that Infinity was going to file for bankruptcy because they couldn't raise enough money to take this forward. So through a series of fortunate events, which we can talk about some other time, we were able to move eganelisib out of the bankruptcy of Infinity into a new biotech company that is now taking it into clinical trials. The company is called Stelexis Biosciences, and I will tell you a little bit more about the trials. And this was just, as a physician scientist, very gratifying that we could be working on something for a couple of years, publish a paper, and then within a year have patients enrolling in a trial. And that was totally facilitated by all the work that Infinity had already done in patients with cancer. So here's one example of that. This is from that phase 1 paper from Infinity of eganelisib in solid tumor patients. And so with this PK data at different dose levels, what they have put on here in the red lines is the IC or the EC90 for PI3 kinase gamma down here. So you'd like to be above that bottom dotted line. And on the top is the EC50 for PI3 kinase delta. You would like to be below that line because you don't want to get into the autoimmune toxicity. And so what is really interesting is that at levels of 30 milligrams and above per day of treating patients, doses that were very well tolerated, we are getting exposure in the steady state above what was sort of the IC50 in sensitive AMLs. And we actually did mouse PK data at the doses that we used in our mouse experiments and showed that it's kind of at similar levels as the human. In fact, maybe the mouse is a little bit worse than the human PK, meaning that if anything, the human exposure will be even better. So what that allows you to do is to go to the FDA and say, look, we have safety data. Instead of doing a phase 1 study where you have to very, very slowly go up from some tiny dose, we can start at a likely biologically active dose. And so this is this current study that is currently enrolling, basically doing a parallel dose escalation in relapsed AML patients and higher risk MDS of single agent eganelisib. And then once the first dose level is cleared, a combination with cytarabine opened as a parallel escalation. So we will get data from patients receiving both single agent and combo cytarabine. The company is exploring those biomarkers, all of them basically, PIK3R5, PIK3CG, phospho-PAK1, phospho-AKT. We're looking at phospho-AKT, just like Infinity had as a PD marker to show that you're getting drug effect on target even in non-tumor cells. And hopefully at some point we'll be able to possibly add biomarker selection and then, simplify the cohorts to a group that be even more likely to respond. So next time I talk, maybe I'll be able to tell you something about the clinical data. We also have a second trial that we're hopefully going to open soon as an investigator-initiated trial led by Jacqueline Garcia here at Dana-Farber, where we can test the venetoclax combination part. And Jacqueline was part of the approval trials for venetoclax to get the ven-aza regimen approved. And so she's an expert in this and has designed a triplet trial, basically, to add eganelisib to ven-aza. She has worked a lot with Tony Latai before he went off to be the NCI director doing BH3 profiling in AML. BH3 profiling, basically dynamic BH3 profiling tests in short-term assays, like in the matter of hours, can predict whether a tumor cell will be sensitive to a drug. And here's proof of concept in a couple of monocytic AMLs that the eganelisib as a single agent maybe is even more effective than venetoclax. And here on the right, you can see that even a little bit more effect when those two are in combination. So we'll be doing a lot of correlative studies as these patients are on trial, both with single agent cytarabine and venetoclax combinations. Okay, I want to just tell you about a few completely new things that are very early. So please, give us any feedback on these. But I think that they are a little bit interesting. So two projects we're currently working on to take things from here forward. So one is, as a physician scientist, I always want to think immediately when we start going into the clinic about what are the mechanisms of resistance and trying to take advantage of patient samples, getting drug, et cetera, to ask that question. So Amanda Fortune in the lab did, traditional, throw some drug in escalating doses onto sensitive AML cells over time and established these 5 parallel, similarly treated parental cells and resistant subclones to eganelisib. And here you can see that the resistant clones are indeed resistant. And then when she did RNA sequencing, you can see that the parental cells have this strong signal for PI3 kinase AKT mTOR signaling, and the resistant cells do not. And when you look at Western blots, I think it's pretty interesting. So these are cells that are among our most sensitive AML cells at baseline. Here in the presence of drug and they're resistant, they have basically shut down AKT phosphorylation at S473 and reduced phospho-PAC1. So again, I don't know what's happening with this AKT phosphorylation, which was sort of much more minor compared to the PAC1 in the short-term assays. But here in the long-term resistant cells, they seem to have shut down signaling, at least by these pathway components. So as you have surmised, we love genome-wide, unbiased CRISPR experiments in the lab. And so with those cells, Amanda has done this experiment. So she put CRISPR library into either the parental cells and then treated them with drug or DMSO, sort of a traditional drug sensitivity resistance experiment, but also then the resistant cells and let them grow in the presence of eganelisib for 14 days and then deconvoluted the data. So we've just gotten the data back. So I'm basically just going to show you the volcano plots of the genes that we found, but I think it's quite interesting. So first, in the parental cells, I would say we got the hits that you would expect with a PI3 kinase inhibitor. So genes on the right are genes that when knocked down conferred resistance to the drug, and genes on the left are genes that when knocked down conferred sensitivity to the drug. So TSC1 and 2 are negative regulators of mTOR. So these being enriched on the right suggests that eganelisib is converging on some mTOR signaling, and that when you sort of artificially activate that, you can make cells resistant to the drug downstream. So whatever it is converging at least there. And I thought this is kind of interesting. PAC2 is essentially the top hit in this screen. And I think that's interesting because of the sort of, again, like I said, discussion about what's downstream of PI3 kinase gamma. We had found PAC1, and the fact that PAC2 loss sensitizes cells to eganelisib tells me that I think this is correct, that probably maybe not just PAC1, but maybe PAC2 can substitute for PAC1 when you inhibit with eganelisib. So we're going to follow this up, but I would say these were kind of not boring, but the expected pathways one might find. The resistant cells were super interesting, and we are just beginning to dig into this. So we saw none of the same pathways in the resistant cells. We did not get top hits that were PI3 kinase, AKT, mTOR signaling. What we found was that I think 3 of the top 7 hits are enzymes in the coenzyme A synthesis pathway. So I didn't know anything about this pathway, but this is a metabolic pathway that starts with vitamin B5 in the extracellular environment being imported into the cell, and via these series of enzymatic steps that require ATP forms coenzyme A. Acetyl-CoA is essential for all cellular metabolism. There's other coenzyme A products that are important for other cellular metabolism. And these resistant cells, resistant egnalcin, are completely dependent on this pathway. You knock these genes out and the cells die immediately. Now, in the parental cells, they do not care. After like 2 or 3 weeks, you start to see a little bit of a fitness defect with knocking these down, but these genes are not like pan dependencies in the DEP map, for example. So this is really an acquired dependency. And at baseline, this is like a colorimetric assay for coenzyme A levels. The resistant cells have an elevated level of coenzyme A compared to the parental cells. So we're still trying to figure out what this all means. Interestingly, Alex Toker, who is at Beth Israel, works on PI3 kinase in cancer, has a paper in Nature 3 or 4 years ago now where they did metabolomics in cells and in breast cancer cells after treatment with PI3 kinase alpha inhibitors. And they also found some interesting changes in this same coenzyme A synthesis pathway and some downstream kinases. So I think there's something real here that we need to figure out. But this will be very interesting to look at in patients who are getting this drug because, again, as I said, it's continuous daily dosing. So the patient's AML cells will be continuously exposed to egnalcin, just like was set up in the experiment here. Okay, now finally, this is your guys' expertise. What's going on with myeloid cells in the tumor microenvironment in leukemia? So this is from that nice review that Jing and Amanda put together in Blood. This is kind of what's known about solid tumors, that PI3 kinase gamma inhibition affects macrophages, maybe dendritic cells. They have an effect on T cells that with immune checkpoint blockade can inhibit tumor cell growth. And we think that PI3 kinase gamma inhibition directly affects leukemia cells. But what about the immune cells in the leukemia bone marrow microenvironment? So this is important not just to kind of understand the parallels or non-parallels, if they are there, between leukemia and solid tumors from this PI3 kinase gamma standpoint, but also because in AML, we try to send all of our patients, if they are young enough and eligible, to an allogeneic stem cell transplant. And in that scenario, we know that the T cell killing of residual AML cells is the reason why allotransplant works. So one wonders if we could actually enhance something about the allotransplant effect on AML with PI3 kinase gamma inhibition. So to understand that, we'd like to know a little bit more about what's going on. So Hailey Ma, who is a postdoc in my lab, has been taking this on, trying to understand AML-associated macrophages in the same way that people have studied tumor-associated macrophages in much more depth, including Jen and Judy and others in solid tumors. And please do not take it out on us for using terminology like M1 and M2, but this is in a paper about AML where they did single-cell RNA sequencing and showed that regardless of what you call them, the type of polarization of macrophages in bone marrows that are also involved with AML is different than in healthy donors, sort of more of these pro-tumor, if you will, M2-like macrophages and less of the M1. And so Hailey decided to try to generate a system that could have AML, more AML-associated like macrophages that we could then do co-culture experiments with AML cells. And interestingly, when people have done this before in the context even of AML, they've usually used interferon and LPS to make primary human monocytes into macrophages. And those are usually more sort of the M1 type of macrophages. And indeed, she found that when you look at protein expression on these macrophages, compared to the interferon gamma, which looked more M1, you can get more, so the closest to what has been reported here as AML-associated like macrophages by using different cytokine cocktails. And she's tested multiple, but IL-4 and 13 are very interesting. They seem to have make these macrophages that are high CD206, CD163, and not as high for things like CD80 and CD86. So then she used those macrophages to co-culture with AML cells with and without eganelisib treatment to ask what happens when you use eganelisib in the AML tumor microenvironment. And she's found a lot of really interesting things. One is that eganelisib treatment, and she's done this in all the ways, treating the AML cells separately from the macrophages and together in co-culture. Essentially, eganelisib treatment of AML and macrophages causes an increase in phagocytosis of those AML cells. She's read this out by flow cytometry and by sort of time-lapse automated microscopy that eganelisib enhances this. Knockout, so genetic confirmation of PIK3CG does the same thing, also enhances phagocytosis of AML cells. So the last slide I have is just a setup of an experiment, but I don't have data to show you yet. But she has now, again, gone back to our favorite unbiased genome-wide CRISPR screens to ask what genes in those AML cells are important for the ability of AML-type associated macrophages to phagocytose them. And she set up this very interesting experiment where she is testing treatment of eganelisib and co-culture with macrophages as sort of a 2x2 interrogation with all the appropriate controls so that she will be able to determine which genes, when knocked down, are important for phagocytosis enhancement or blocking, independent of the effect of the eganelisib on the AML cells alone. So she can do, a lot of different comparisons from this. And I will say that early data look very encouraging. And the next time you invite me to speak, I'll let you know what we found from that. So this is the end. I think we found that PI3 kinase gamma inhibition is active as single agent in AML, enriched for leukemias with monocytic and dendritic differentiation, but not absolute. The single agent activity seems highest in these PIK3R5 phospho-PAC1 high leukemias. We've done a little bit of work on what's upstream and downstream, and I think we're still trying to think about that in more detail. We have some idea of which combinations might be most active in AML, and we think that the microenvironmental effects in the setting of AML definitely need more attention, and that's something we're working on a lot. So the things that we're doing now are pushing these clinical trials, trying to refine biomarkers in the setting of trials and in the lab. We're very excited to be able to study actual leukemias from patients on trial to look at some of these same pathways, to figure out mechanisms of resistance, and then hopefully we'll figure out how to use this to enhance AML cell phagocytosis or some other in-trans effect. We didn't even start to explore the sort of cytokine polarization effects that were also implicated in the solid tumor setting. So I think I mentioned the people who are doing most of the work here, and here are their names again, especially the ones in purple, done a lot on this project, and I think we have some time for a few questions. Thanks.
Well, thank you for an amazingly impressive talk. I would say this may be one of the most comprehensive talks we've had in quite a while, going from discovery to clinical trial and back to resistance mechanisms. If anybody has any questions, please feel free to ask them directly. I would like to ask a question, and I think Jelani and Jennifer can handle other questions.
Yeah,
Maybe
So-
stop sharing so that we can all see each other. There we go. Okay.
Yeah, there we go. Well, I have a million questions, but so are there cell lines like FLT3 ITD mutations that are completely resistant to PI3 kinase gamma inhibition, or I don't mean cell lines, but I mean tumor types?
Tumor types that are completely resistant. So there are AML cells, both cell lines and PDXs, that are completely resistant. But it doesn't seem to correlate with any of those genetics. For example, we didn't, in the paper from Duke and Paris, they used MOLM13 cells quite a lot as sort of their target cell. And that has a FLT3 ITD mutation. So
Yeah.
in our hands, that cell line is only kind of like an intermediate sensitivity cell line, actually. So but it's not that having the FLT3 signaling pathway active completely abrogates the ability of blocking gamma to do anything. So I think your question is like, even with an obvious different kinase active mutation, you don't completely bypass this pathway. Now, I'm not an expert in the other cell types. In AML, for sure, there's a lot of known interaction between all these pathways, FLT3, RAS, now I think more appreciation PI3 kinase, and it's not always clear what's upstream of one and downstream of the other one. But yeah, we haven't found any kinase that confers absolute resistance.
Okay, and then one more question I had is, do you see upregulation of PI3 kinase delta as a resistance mechanism?
Yeah, that is a good question. We did not see it at the RNA level. In those resistant cell lines that I showed you, we did DNA sequencing, first of all, to make sure there wasn't like some commonly mutated RAS or something as a bypass. There was not. I should have mentioned that. At the RNA level, we did not see another PI3 kinase specific gene, but we did not do even Western blots for delta or any of the delta components. I don't know, well, you tell me, but with that phospho-AKT being just completely gone in the resistant cells, it makes me think that we're not going to see like alpha, beta, or delta being the compensatory pathway, but we probably should look at that.
Yeah, and it may depend on cell type, mutation, combinations. Yeah.
Yeah.
Okay. Yeah, thanks.
Judy, you want to keep going or should we pass it along? I think Jelani, do you have a question?
Yes, I do. So, Andy, fantastic talk. I had a question actually about those resistant cell lines, those OML, OCL, AML3s. Did you look at threonine 30? I noticed that serine 473 was analyzed, and the resistant cell lines had no phosphorylation of serine 473. But I was wondering about the PDK1 to threonine 308 signaling in those cell lines. Do you all see that abrogated as well?
I don't believe we tested it in those resistant cell lines yet. We probably should. I mean, we've taken you can either or both. You can go after like everything that's published about PI3 kinase and test like individual pathways, which gives us a lot of information. Or, we say, let's just go to our broad genome-wide screen and go for something that seems interesting. But I think at some point we'll probably have to look at that too. 473 was the one that was most affected in our original work in the phosphoproteomics, and that's why we've gone after that one. But if you reviewed our paper, you also asked us to look at 308, which we did, and so it's in the paper for all the original stuff, but we haven't done it in the resistant cells yet.
Right.
Let's see, sorry, I'm butchering your name. There's a question in the chat. Do you want to ask your question or should I? Okay. Andy, I'm sure you can read it too, but phagocytosis is a normal homeostasis biological mechanism. Clear the waste cells and how do, sorry, I have a box over it. I can't get rid of this. There we go. Agnosilibab, phagocytate normal healthy donor 1 and 2 that you have shown.
Yes. So, well, I would say we are working out in more detail how to isolate, let's say, the effects of PI3 kinase gamma inhibition on the AML cell and the macrophage, which is right next to it. So that's already a very good question, because in the solid tumor world, we're going to talk about inhibiting the macrophage, PI3 kinase gamma, and the breast cancer cell next to it doesn't care about that pathway, so it's irrelevant. But here, the drug, and if you're giving a drug to a person, it's going to affect both of those cells. But we did the screen in the AML cells. So we are looking for pathways that modulate the ability to be phagocytosed when they are blocked, knockdown or, vice versa in the AML cells themselves. We may also try to knock some things down in the primary macrophages. That's harder. We're trying to avoid a situation where we use an AML cell line as the macrophages, which people do in sort of non-AML work. But I think here we want to try to avoid that. The other part of your question you might be asking is that we are now also testing this in that AML cell line in which we did the screen is one that is sensitive to eganelisib, but we used eganelisib at doses that does not overtly kill all the AML cells. So we're also now going to test that ability of eganelisib to increase AML cell phagocytosis. Is that also relevant in an AML cell that doesn't die when you treat it with eganelisib? So it could be, back to kind of Judy's question, that could there be AML cells that where eganelisib would be useful in vivo, even if it doesn't directly inhibit their growth, by doing something in the AML cell and or in the adjacent macrophage that encourages their phagocytosis. I didn't have time to get into this, but the CD47 blockade story is something that's been important across cancer, but in AML, that's where it was sort of first discovered, if you will. It turns out that there's probably some species-dependent differences there that drove a lot of those phenotypes, but there's still a lot of interest in having AML cells be phagocytosed by macrophages. So even if the gamma inhibition part doesn't directly affect the AML cell, either because it just doesn't work in AML or doesn't work in all AMLs, or they're more resistant ones, I think it still could be interesting to see how it's affecting the adjacent macrophages.
A similar question is from Kent Lin, but related to did you see an increase in MHC presentation, which may be more relevant to AML pathology?
We did not. Yeah, as I said, we only looked at phagocytosis so far. And I would say actually not just phagocytosis. We also, there are some genes in our CRISPR screen. So in our CRISPR screen, highly analyzed both the AML cells and what changes in the AML representation after their co-culture with macrophages, and then also analyze the macrophages themselves. So an AML cell that gets eaten by the macrophage has now the guide RNA inside the macrophage. And you can actually very clearly see that genes that affect phagocytosis, and other people have seen this in their screens as well, are kind of on this diagonal, if you will. So they go up in the AML and they go down in the macrophages, or vice versa. There are also genes that only seem to affect AML cell viability in the AML cells, but aren't phagocytosed, but that still only happens in the presence of macrophages. So this is not directly your question, but it could be that cytokines or something else that's being elaborated by the macrophages affects AML cell viability, independent of its ability to phagocytose them. The last thing I will say is that in our paper that's published in all of Chingyu's work, unlike most things, it always seemed like the in vivo experiments actually were a little bit even more impressive than the in vitro experiments. In vitro, often we're seeing like cell cycle arrest, a little bit of apoptosis. But in vivo, to see those survival differences, I think there has to be something more going on. And one possibility, of course, because NSG mice have macrophages, is that there's something going on there in the tumor microenvironment, maybe independent of T cells. That's another reason why we kind of went after phagocytosis and not the sort of adaptive immune stuff so far.
Judy, did you want to ask any follow-up
questions?
Oh,
yeah, I have a question. So a lot of people don't know this, but the first observation we made with inhibiting PI3 kinase gamma in myeloid cells is that it regulates the trafficking of monocytes and granulocytes and probably dendritic cells into tumors and inflammatory tissues. So I know that one of the effects that were studied with PI3 kinase delta gamma inhibitors like duvelisib was whether it caused release of leukemia cells into circulation and made them more vulnerable to chemotherapy. So do you see any effects on AML cells related to survival? Because there was this, there were a long time ago observations about AML interacting with stromal cells in the bone marrow, and that the interaction was integrin alpha 4 dependent. And then if you blocked alpha 4, you could cause sensitivity to chemotherapy. And so PI3 gamma in primary myeloid cells regulates alpha-4 activity. So if you block it, alpha-4 is inactive and cells can't adhere. So have you seen any observations related to that, or
Super interesting. Well, I would say we have not directly looked at it. We'll see. Obviously, in the clinical trial, it'll be interesting also if we see like an increase in circulating AML cells or something on
yeah,
therapy. In the mouse, it's hard to do like bleeds, peripheral blood bleeds in the mouse and you'd have to really design an experiment to specifically look at that. But certainly you are correct that AML has an important interaction with the bone marrow microenvironment. And there are like immune exclusionary zones even in the bone marrow with AML. We think about them in solid tumors, but it's also, there's this hypoxic niche that's next to the bone and next to certain vascular cells. AML lives there. And one wonders if, maybe we release them and they become more susceptible to immune cells or to chemotherapy or whatever. I mean, there were trials back in the day using things like GCSF even, or interferon in AML that releases the AML cells and supposedly makes them more sensitive to chemotherapy. So that's fascinating. Specifically, there is this paper that just came out I think it's in iSciences, that was looking at AML homing, and there was they did look at this pathway, including PIK3R5 sort of being a hub that you're nodding. Maybe this is your paper. I hope it's not. That would be very embarrassing.
No,
but anyway,
no.
If maybe you know the paper, but showing that indeed this pathway is important for AML cells' ability to interact with its microenvironment. So I think that's also, it would be great if there's multiple ways that this drug can work.
Yeah. Well, maybe in your CRISPR screens, you might have something that comes out of that related to adhesion, migration. That might be interesting.
Totally. Does that integrin pathway affect phagocytosis at all by macrophages? I don't know.
So here's the interesting. PI3 gamma definitely regulates membrane remodeling. So it regulates macropinocytosis, and there are a couple papers that show that it interacts with RAC to regulate phagocytosis. And your stuff around PAC1 and RAC and PI3 gamma kind of makes sense. It's not a highly studied pathway. We haven't done anything in that. So I think that's could be insightful about regular myeloid biology too, but it might be important here.
Yeah, that's great. I will look at that. We've looked at those papers are mostly in the macrophages, looking at how gamma pathway affects macropinocytosis, et cetera, et cetera. And we're trying to see what it does in the AML side, but it could be both. I mean, I think it's known that sort of membrane physical biology is very important in phagocytosis. So it probably might be important on both sides.
Yeah, and they could all be related to many processes like migration, metabolism, a lot of ways. So it's fascinating, but really terrific work. Really exciting to see such well characterized work leading to clinical trials and hopefully saving people.
We hope. Well, thanks again.
Thank
Inspired by your work for sure.
All right. Well, thank you so much, Andy. And for those that are still on behalf of the Myeloid Network, happy holidays, happy new year. We'll pick up again January 8th with Doug Hanahan. So great to see everyone. Thanks so much, Andy. It was brilliant.
Bye.
Thank you. Yeah, thank you.
Bye, Judy. Bye, Jelani.
Bye.